Hey there! Let's dive into the fascinating world of bone repair and the crucial role played by a family of proteins called sirtuins. Our bones have an incredible ability to regenerate and heal themselves after injury. But this process is complex and can be disrupted by various metabolic and health conditions. In the United States alone, around 100,000 fracture patients experience non-union annually, which is a significant health concern. Enter the sirtuins, a group of enzymes that have gained attention for their potential to regulate bone metabolism and repair. These enzymes, expressed across different tissues, act as histone deacetylases and adenosine diphosphate (ADP) ribosyl transferases, and they play a key role in cellular energy metabolism and signaling pathways related to bone remodeling. Recent research has highlighted the importance of glucose metabolism in the function of osteoblasts and osteoclasts, the two main cell types involved in bone repair. Both cell types have high energy demands, and glucose serves as their primary energy source. Disruptions in glucose metabolism can impair bone cell activity and hinder the delicate balance between bone formation and resorption. This review aims to explore the role of sirtuins in bone repair, specifically how they influence glucose metabolic pathways and energy signaling in osteoblasts and osteoclasts. By understanding these regulatory mechanisms, we can potentially develop targeted therapies for metabolic bone disorders and improve fracture healing. The search strategy for this review was comprehensive, covering multiple databases and including both English and Chinese language publications. The review focused on specific sirtuin isoforms such as Sirt1, Sirt3, and Sirt6, which have well-established roles in bone metabolism. The review also delves into the complex interplay between osteoblasts and osteoclasts, the two key cell types involved in bone repair. Osteoblasts, which originate from mesenchymal stem cells, are responsible for bone formation, while osteoclasts, derived from hematopoietic stem cells, drive bone resorption. The balance between these two cell types is crucial for successful bone remodeling and repair. The role of glucose metabolism in bone cells is a key focus of this review. Osteoblasts, for example, rely heavily on glycolysis, a metabolic process similar to the Warburg effect seen in cancer cells. This metabolic preference supports the biosynthetic demands of collagen production and extracellular matrix mineralization. On the other hand, osteoclasts, which require significant energy for bone resorption, shift towards glycolysis as they mature. Sirtuins, particularly Sirt1, Sirt3, and Sirt6, are known to participate in the glucose metabolism of bone cells. Sirt1, for instance, has a dual regulatory role in osteoblast and osteoclast differentiation and can influence bone metabolism in both the skull and long bones. Sirt3, on the other hand, is primarily involved in regulating mitochondrial energy metabolism in bone cells, supporting bone homeostasis. The review also discusses the potential of targeted therapeutic approaches, such as sirtuin activating compounds, SACs, and sirtuin inhibitors, for treating metabolic bone diseases. While these targeted drugs show promise, further research and clinical trials are needed to fully understand their long-term safety and effectiveness. In conclusion, sirtuins offer a potential pathway to explore bone repair mechanisms and develop new therapeutic strategies for metabolic bone disorders. By understanding the intricate relationship between sirtuins, glucose metabolism, and bone cell function, we can work towards improving fracture healing and maintaining bone health.